Morning, everyone. So it's my great honor to be here as a keynote speaker. So my name is Xujun Duan, and I am from the University of Electronic Science and Technology of China. So today I'm going to share with you the neuroimaging studies of ASD for the almost the past decades of our lab. And my title today is the mapping brain functional and structural differences in ASD, moving towards precision treatment. I have no financial interests or relationships to disclose. What is ASD? ASD is autism spectrum disorder and is a neurodevelopmental condition that leads to communication, social, and behavioral challenges. The ASD prevalence has grown dramatically from the past decades. And according to the data released by CDC in 2023, 1 in 6 children had a diagnosis of ASD in US, and the estimated prevalence of ASD in China is around 1%. Although the neurodiversity perspective has been well acknowledged and respected, we should still keep in mind that there are still 32% of the ASD people have intellectual disability, and families on this severe end of the spectrum often struggle for support and treatment. So what causes ASD? Research suggests that ASD develops from a combination of genetic and non-genetic or the environmental influence. But how do this genetic and non-genetic influence give rise to ASD? I guess most appear to affect crucial aspects of early brain development. Previous studies has demonstrated brain anatomy related to cause importance in ASD, especially the brain regions of orbital frontal cortex, the medial prefrontal cortex, fusiform gyrus, amygdala, insula, and their relationship with their anatomy, functional, and connectivity with social deficits in ASD. DiMartino and colleagues also demonstrate hyperconnectivity in local connectivity and hypoconnectivity in long-range connectivity in ASD. And Supuka and colleagues demonstrate that those abnormal connectivity in ASD might result from the abnormal neural fluctuations. Although these studies contribute greatly for understanding the brain biology in ASD, ongoing debate exists within the neuroimaging literature. And inconsistent findings attribute to difference in demography, heterogeneity in differences in demography and heterogeneity in clinical manifestations, scanner parameters, or the methodology of data analysis. Except those factors, the dynamic nature and developmental changes of the brain should also take into consideration. And differences have been observed at group level mostly, and thus ignore the individual differences. So we need to also take the heterogeneity into consideration. And another concern is that over the past decades, great efforts have been made, and how can we advance neuroimaging to clinical practice? So over the past several years, we have dedicated to delineate a mapping of the different structure and function of the autistic brain, and our goals can be fall into these 4 sections: mechanism, prediction, classification, and clinical implications. So that's what I'm going to share with you today. The first session is mechanism. In this session, we aim to understand atypical brain networks of ASD by using resting state fMRI. So ASD is a disorder of brain network organization. And resting state fMRI has emerged as a powerful tool to assess the intrinsic brain activation of the brain, intrinsic organization of the brain. Over the past decades, resting state fMRI studies have reported disrupted brain connectivity in diverse systems in individuals with ASD. However, inconsistent results in functional connectivity were reported. Resting state functional connectivity was defined as the temporal correlation between remote brain regions. Most of previous studies assessing resting state functional connectivity based on the assumption that the connectivity patterns remain constant over time. But theoretical models and empirical observations suggest that the functional connectivity pattern changes dynamically in relation to the ongoing rhythmic activity of the brain. So capturing the nuanced time-varying properties might advance our understanding of differences in functional connectivity of ASD. So the first study that I'm going to share with you is the dynamic connectivity of the right anterior insula in ASD. The right anterior insula plays an important role in social connection, and as a critical hub of the silence network, it was proposed to act as a hub in initiating the switching between the internally and externally focused networks like the deformal network and the frontal-parietal network during cognitively demanding tasks and the resting states. So in this study, we utilize the open access ABIDE dataset to assess the dynamic properties of the anterior insular functional connectivity and its association with social deficits in ASD. By using the sliding time window method correlation, we found that this region has various connectivity pattern, and it was positively correlated with the deformal network in some states, but negatively correlated with it in another states, demonstrating the flexibility of this region in connectivity with the task negative network and the task positive networks. And the next step is to assess whether altered connectivity in this region can be detected in all the states in ASD. As we can see here, not every state exhibits altered functional connectivity in ASD, but only in stage 3 and stage 5. Decreased functional connectivity between the right anterior insular cortex and the medial prefrontal cortex was found in ASD compared with TD. And by using meta-analysis, we found that stage 3 and stage 5 were related with cognitive processing of social, mentalizing, self-referential, and theory of mind, indicating that altered functional connectivity of the anterior insula was state-dependent. And the medial prefrontal cortex is a core region of the deformal network and also a core region of the social brain because of its important role in self-referential and social processing. So, previous studies based on static functional connectivity analysis have demonstrated abnormal inter- and interhemispheric pathway in ASD. And by taking advantage of functional connectivity density and the sliding window analysis, we demonstrate that both inter- and interhemispheric corrections show aberrant dynamic functional connectivity density variability in the social brain regions, including the medial prefrontal cortex, the fusiform gyrus, and the temporal pole in individuals with ASD compared with TD. And all these regions, again, fall into the social brain. From one single brain region to the inter- and interhemispheric, and we also assess the whole brain networks in ASD. By constructing the whole brain dynamic networks, we observe that an overall hypervariant dynamic connectivity in ASD, indicating the unstable connections all over the brain. And the most unstable connections were related with the medial prefrontal cortex and the temporal pole, and also the social brain regions. We also review changes of different connectivity patterns across the states, and we demonstrate a state-dependent alteration of functional connectivity in ASD. So the brain state might be one of the factors that leads to inconsistent results of the previous resting state functional connectivity literature. From different network level, we consistently observe the differences in brain connectivity of the social brain regions. The social brain is responsible for social connection and social behavior, and it includes the amygdala, the medial prefrontal cortex, the superior temporal circuits, the insula, the temporal lobe, and the fusiform gyrus. So, in order to evaluate the dynamic interaction among the social brain regions, we employ multivariate gradual causality to assess the intrinsic effective connectivity within the social brain of ASD. And we found that significantly attenuated effective connectivity from the medial prefrontal cortex and the bilateral amygdala in children with ASD compared with TD. And the medial prefrontal cortex provide contextual and experimental inputs to the amygdala to regulate emotional responses triggered by the amygdala. And the attenuated MPFC amygdala pathway in the social brain might related to the impaired modulation of the prefrontal cortex to the amygdala in regulating social emotion responses, thus resulting the social behavioral challenges in ASD. So, by capturing the dynamic properties of the functional networks in ASD, we reach a consensus that the social brain are the most effective regions in the autistic brain. Another important factor that leads to the inconsistent finding is the developmental change of the brain. Depicting the developmental change or developmental trajectory might provide insights into how brain anatomy and function differ across lifespan. Because ASD undergoes an atypical trajectory of brain maturation, and a developmental model with hyperconnectivity in young children and hypoconnectivity in adolescents and adults was proposed by Dr. Wooding, highlighting the importance of investigating the neuroetiology of ASD from a developmental perspective. So in this session, we use multimodal brain imaging data to predict the brain developmental trajectory of ASD. Age estimation obtained from brain kinetomics might reflect the level of brain maturation along the neurodevelopment. Here, we estimate the brain kinetome age from structure functional kinetomics constructed by white matter fiber tracking and resting state functional connectivity. We found that brain kinetome age was greater than the chronological age in childhood ASD, but the brain anatomical age was younger than the chronological age in the adolescents with ASD. And the brain kinetome age matched well with the chronological age in TD children and adolescents. And by summarizing this data, we suggest that ASD might exhibit accelerated brain maturation in childhood, followed by a delay starting at pre-adolescence. So in previous study, we demonstrate a shift from the acceleration to delay maturation at pre-adolescence. But at which particular age this shift take place, and whether different brain networks undergo distinct developmental causes pertaining to the timing at delay maturation emerges. To answer this question, we construct 6 resting state networks, 3 primary networks, and 3 high-order networks: the deformal network, the central executive network, and the silence network. By using a curve-fitting method, we delineate the developmental trajectory of the 6 intrinsic networks in ASD and TD. So red is for ASD and black is for TD. We can see an interaction between the developmental curves of ASD and TD for those 6 brain networks. And there is a shift from hyperconnectivity to hypoconnectivity at the intersection points. And by using a sliding threshold method, we demonstrate an age range for each network, within which each network shifts from the hyper to hypoconnectivity. We also found that this shift of the primary networks took place earlier than the high-order networks. For example, the shift from hyperconnectivity to hypoconnectivity happened of the deformal network takes place between age 11.5 to 13.5, indicating that if your subjects fall into this age range, you might not be able to detect the differences in deformal network between ASD and TD. Because ASD is an early onset neurodevelopmental condition, data from younger participants are very important to delineate the developmental changes in early childhood ASD. In this study, we recruit young children aged between 4 to 7 years old, and by using development predictive modeling based on 4 Y-matter index, we estimate the Y-matter abnormal development index of children with ASD and identify 2 subgroups of ASD using the K-means clustering method. We found an inverse pattern of atypical Y-matter microstructure in these 2 subgroups, and also different clinical manifestation in these 2 groups, and demonstrating 2 different developmental stage, with stage 1 showing overgrowth with weak, weak social deficits or weak symptom severity, and the stage 2 exhibit delayed maturation with severe clinical symptoms. So from previous 2 sections, we can see that ASD exhibit high-level heterogeneity in brain imaging, and traditional case control analysis might not be able to capture the individual differences in ASD. So in this session, I'm going to talk about the heterogeneity and subtyping of brain structure and functional data of ASD. So individuals with ASD generally exhibit multi-level heterogeneity across genetics, brain systems, and clinical manifestation, which has been the largest obstacle towards the understanding and treatment of ASD. And the identification of homogeneous subgroups of ASD may help depict this heterogeneity, thus facilitating diagnosis and individual treatment. And in previous studies, the features used for subtyping were either based on prior knowledge or high-dimensional data, or match normal controls rather than quantitatively measurement in the at the individual level. So in this study, we calculate individual neuroanatomical difference patterns for each ASD by assessing the difference between each participant with ASD and 20 TD from the same sites with the most similar age and FIQ to the corresponding ASD participant. And then clustering methods was next used to stratify individuals with ASD into 3 subtypes with different gray matter patterns, one with the decreased gray matter volume, another with increased gray matter volume, and the third subtype with a mix of decreased and increased gray matter volume. And we also find different clinical symptom severity and functional connectivity among those 3 subtypes. So this study show a way to compute an individual structural difference map by matching a group of most similar, typically developing controls for each individual with ASD. However, the generalizability and clinical use of the approach is limited due to the matching strategy. So, to develop a parameterized, low-dimensional, and individualized metric to delineate homogeneity of brain structure in ASD, we use the non-negative matrix factorization to decompose the whole brain gray matter into distinct factors and their corresponding weights, and then use normative modeling to estimate the factor deviation. The normative model is similar to the height and weight development curve used in pediatrics, which provides statistical inference to delineate the individual difference on quantitative biological measures. By normative modeling, we can also draw a developmental chart for the brain and assess the deviation of each individual with ASD. If we want to draw a developmental chart, which brain region should be targeted? Since the whole brain contains more than 100 brain regions, right? So dimension reduction is an important step. The non-negative matrix factorization has recently emerged as a promising pathway to capture robust and reliable non-negative regional differences in high dimensional data. So by using the non-negative matrix factorization, a matrix can be decomposed into a factor matrix and a weight matrix. The gray matter map of a single subject can thus be represented as a linear combination of the basic factors. And in this study, by using non-negative matrix factorization, 6 latent gray matter factors and their corresponding weights were obtained. And those 6 factors, like the factor 1 represents the medial cortex, factor 2 represents the cerebellum, factor 3 represents the perceptual regions, and then factor 4 for the parietal region, factor 5 the frontal region, and factor 6 for the limbic system. And after obtaining the weights of factors from TD gray matter matrix, we develop a normative model to draw the normative developmental charts based on the weights of the gray matter factors. And individual deviation of the 6 latent factors were then obtained for ASD, and finally, 3 subtypes with distinct neuroanatomical deviation patterns were identified by clustering methods, and among which distinct clinical manifestations in social communication deficits were identified among those 3 subtypes. And the previous study demonstrated the heterogeneity in structure brain. So individuals with ASD also exhibit high-level heterogeneity in functional connectivity. And case control comparison ignore how ASD individuals differ from another in terms of underlying biology, and also fail to appreciate heterogeneous ASD-related functional connectivity abnormalities. And here, Tang and colleagues imply a decomposed resting state functional connectivity of individual with ASD into multiple abnormal functional connectivity patterns with this different whole brain hyper and hyper resting state functional connectivity patterns. And those studies demonstrated homogeneous subgroups, but individual share and individual specific subspace of altered functional connectivity in ASD remains unknown. So, in this study, we aim to project a typical functional connectivity of ASD onto an individual share and individual specific subspace by using common orthogonal basis extraction. So this algorithm provides an approach for decomposing multi-block data into a common subspace and a block-specific subspace. And by using this method, 3 components were reviewed for the individual share spaces of the based on the atypical functional connectivity in ASD. We then examined the association between components of this individual share and individual share subspace of atypical functional connectivity at the functional connectivity differences at the group level, which is the between group differences in functional connectivity. Okay, we also investigate the relationship between the weights of the individual share and subspace and the clinical importance. We found that all 3 components of the individual common subspace was significantly correlated with the functional connectivity differences at the group level. Okay, at the group level, at the between group differences. Further, the correlation analysis showed that weights of common components were not associated with clinical importance. So what is so if the common space is not related with the individual differences, so how about the individual specific subspace? So we further look at the correlation between functional connectivity in the individual specific subspace, and we found that altered functional connectivity of this specific subspace within the especially for the deformal network and the CON and the FPN was significantly correlated with the cause importance in ASD. And further, we found that by using atypical functional connectivity in the individual specific subspace can improve the prediction of importance severity compared with that use the functional connectivity or abnormal functional connectivity of the between group differences. So we found that individual share subspace in the atypical functional connectivity of the individual share subspace reflects differences at the group level, which indicating the between group differences. And while the individual specific subspace represents the individual variation in clinical importance. So the previous sections demonstrate the difference in brain networks, developmental trajectory, and heterogeneity in ASD. How can we advance neuroimaging to clinical practice? So for ASD treatment, we should note that there is no evidence for treatments that claim to result in cures or recovery from autism. And because young children with ASD have impairments or challenges in areas of communication, social interaction, and often display repetitive on-like behavior, and this early pattern of difficulties is a challenge for parents. Therefore, approaches that help parents develop strategy for interaction and management of behavior are the obvious routes for all early intervention in ASD. Like the areas to target in intervention is like communication skills, engagement, motivation, attention, imitation, social skills, and play, and so on. And for behavioral therapists, previous study has demonstrated social skills interventions have been found to improve social competence and understanding, and this kind of behavioral therapy should be the earlier the better. And this study by both demonstrate that cognitive training can induce plasticity of the social brain in ASD. So we can see that after cognitive training, the activity within the social brain, especially for the fusiform gyrus and the amygdala, increase significantly after training. Since the neuromodulation method or technique can modulate the brain functioning and results in neuroplasticity, we are wondering whether it can generate similar effects of cognitive training. As a commonly used neuromodulation technique, the transcranial magnetic stimulation, we call TMS for short, has been widely used for treatment in neurological or psychiatry disorders. And it's a non-invasive brain stimulation technique and it can result in altered cortical excitability. In this review by Oberman and colleagues, they proposed that the TMS might be a promising emerging tool for potential treatment of ASD, but the available literature on the TMS use in ASD is very preliminary, and the challenges is where to stimulate and how to stimulate. Based on the social brain theory of ASD and the consistent findings of the social brain network dysfunction in ASD from our studies, we suggest that the social brain network can serve as a biomarker and targets for intervention in individuals with ASD. The amygdala is a central component of the social brain and plays a crucial role in social emotional processing. In our previous study, we found that the amygdala and its connectivity with prefrontal cortex might directly contribute to the social challenges observed in ASD. However, the amygdala was located deep within the temporal lobe and it cannot be directly stimulated by non-invasive TMS techniques. But prior research indicated that non-invasive stimulation on the scalp is most effective when targeting a region exhibiting strong connectivity with the optimal sites for deep brain stimulation. And because the amygdala is used to be a target of the deep brain stimulation, for very severe ASD previously. We developed a personalized amygdala-optimized functional connectivity guided TMS strategy in which we target the amygdala by stimulating the prefrontal cortex region that display the strongest connection with the amygdala. This approach enables us to individually target the amygdala and address challenges associated with the targeting this deep brain region. This is the personal TMS target by this method. And after several times of stimulation, we obtain feedback from caregivers like they claim that after stimulation, there has enhanced sleep quality and increased duration of sleep and increased interaction and eye contact, increased pro-social behavior, and enhanced emotional stability of the children. So this kind of small progress greatly delighted the families and encouraged us. But how is the scientific data? So we imply a randomized double-blind control design in which children with ASD aged 2 to 8 years were assigned to either the amygdala-optimized functional connectivity group or a non-optimized control group. So participants in the amygdala group receive personalized amygdala targets cTBS stimulation, while the non-optimized control group underwent the same stimulation protocol, but with stimulation site determined use the canonical EEG 10-20 system of the F3 sites, which represent the classical dorsal prefrontal cortex, the left dorsal prefrontal cortex. So here we use the cTBS, which is the continuous theta-burst stimulation, and it's a type of repetitive TMS, which has the advantages of shorter stimulation time and longer lasting after effects, making it particularly advantageous for pediatric population due to their increased tolerability and reduced stimulation duration. And we assess the clinical effects of the cTBS using the ADOS, which is a standardized measure of clinical phenotypic feature associated with ASD. We found that the amygdala group demonstrates significantly reduction in the ADOS scales, especially for the social interaction, the social communication, and the total score. In addition to the ADOS, we also examined the cTBS-induced changes on several parents' rating behavioral measurements, like the autism treatment evaluation checklist, the child behavioral checklist, the repetitive behavioral scale revises, and so on. We found that the amygdala group demonstrate significant reduction in scores on most of the scores. We also assessed the changes in eye tracking data before and after the treatment. We found that after the treatment, there was a significantly increase in gaze fixation on face and eye regions in the social picture in the amygdala group. But for the control group, there was no significant differences in eye tracking data before and after the treatment. To determine whether cTBS stimulation induced structural changes in the amygdala, because we want to target the amygdala, we examine changes in amygdala gray matter volume using high-resolution MR data and free surface segmentation of brain structures. And we also evaluate the amplitude of the low-frequency fluctuations to detect the local functional changes of the amygdala after the treatment. We found that after treatment, there was a significant decrease in volume and in spontaneous neural reactivity of the right amygdala in the amygdala group. And there's no such changes in the control group before and after the treatment. So we would like to look at whether those changes in volume, amygdala volume or activity or fluctuation is related with this important improvement, but we do not find any correlation because we think that the amygdala might, the behavioral changes might not dependent on the amygdala itself. It might depend on the widespread connectivity between amygdala and the brain networks. So we examine functional connectivity between bilateral amygdala and 7 intrinsic functional networks in the amygdala group. And we found that changes in the right amygdala functional connectivity with the dorsal attention network, frontal parietal network, and the deformal network has significantly correlated with the reduction rates of the after the treatment. Okay, so wait a minute. Okay, so in the control group, we retrospectively compute optimal stimulation targets for each participant and baseline based on their amygdala functional connectivity maps. We found that stronger connectivity and closer spatial proximity between the actual target and the optimal target within the control group are significantly correlated with greater ADOS score reduction. So this study demonstrates that the personalized amygdala-optimized functional connectivity guided cTBS can effectively improve core phenotypic symptoms of autism in young children at ages closer to clinical diagnosis. So over the past several years, we have concentrated on mapping the brain structure and functional differences in ASD. And in this process, we built up a large brain imaging data set of Chinese autistic children and filled the gaps of lacking brain imaging data of Chinese autistic children, especially for young ages. We also established an assessment intervention imaging data platform. And on this platform, we have developed the brain imaging-based subtyping software, the eye tracking-based ASD assessment software, and personalized target generation software. And we also generate quantitative eye tracking and neuroimaging assessment reports for ASD, especially the comparison before and after the treatment. So as you can see, although we have recruited our own data, but most of our previous work was still based on the excellent open access biodata sets due to the small sample of our own data sets. And we have no idea on the structural and functional properties of the Chinese children with ASD based on large sample. So therefore, I have initiated the China Autism Brain Imaging Consortium to promote the brain imaging studies of ASD in China to a large sample, multi-site, open science one. Right now, we already have 9 sites joined in, and the sample size is over 1,600. And our first goal is to draw a brain growth charts of the Chinese ASD children for a better understanding of the neuropathology of this condition and probably provide better supports for this population. So in the end, I would like to thank all of my collaborators, especially my PhD supervisor, Hua Fuchen, in my university, and also Dr. Vinod Menon at Stanford University. And he is also my supervisor when I was at Stanford. And also I would like to thank Dr. Lucina Wooding. We have known each other for more than 10 years, although during this period of time we have barely seen each other in person, but lots of the work study that I have talked today, is collaborated with Lucina. And I would like to thank all of my collaborators in China, and most of them are pediatric doctors. And this is my lab. Most of the study was conducted by them, and we are very excellent. We are a very motivative and excellent research team that we would like to really contribute to the understanding of ASD and finally the treatment of ASD. Okay, thank you. That's all.